Hello students, now that you have read about cytoskeleton's structure and function, including its definition, let's look at some of the cytoskeleton disorders. So in this mini lecture, I'm going to present two cytoskeleton disorder. The first one is hereditary spherocytosis. To understand this disease, firstly, we must understand the structure of red blood cell. When we talk about red blood cell, the red blood cells are exposed to high amount of pressure and force because they navigate through the narrow capillaries in our body. Okay, so in order for them to squeeze themselves through this narrow capillary, they undergo rapid and reversible deformation. All right, so they are deformed and then they form back. All right, to cope with this stress, the red blood cell has a specialized cytoskeleton. All right, so the system is very special in a way that it provides the mechanical stability and flexibility that is needed by the red blood cells to withstand the forces they experience during their navigation or circulation in the body. Okay, so in terms of the structure, the red blood cell has a lipid bilayer, a transmembrane protein, and a filamentous meshwork of protein. So the filamentous meshwork of protein, this is what forms the membrane skeleton along the entire cytoplasmic surface of the membrane. Okay, and the most abundant protein, all right, in the membrane skeleton is pectin. Let me explain using a diagram. Okay, so the first picture you see here, this is red blood cell, the biconcave shape. All right, and as you zoom the surface, you can see that this is how it looks. You can see the mesh work of protein here. And if you further zoom, you can see the network more clearly. All right, and if you further zoom this is illustration here. You can see that this is the transmembrane protein in the middle, and then it is supported by the alpha and beta spectrin, all right, which is the membrane skeleton that made of meshwork of protein. Now let's look at what happens in hereditary spherocytosis. What happens is predominantly a process called hemolysis. Hemolysis meaning the red blood cell is being lysed or destroyed. Now let's look at picture A. This is a normal erythrocyte in biconcave shape. If you to take out a small portion of this, and then when you zoom, you can see what is the structure like. Here you have membrane, and then here you have cytoskeleton. All right, the interaction between the membrane and cytoskeleton is supported by transmembrane protein that you see here. The transmembrane proteins are made of various different proteins such as band 3, band 4.2, ankyrin, and here you can see glycophorin, actin, and so on. All right, so this interaction between membrane and also cytoskeleton mediated by the transmembrane protein provides flexibility and mechanical support to the red blood cell when they go through circulation. All right, what happened in spherocytosis is that's mutation. All right, so this mutation will cause deficiency of certain proteins as what you see shaded here. Therefore, there's a disruption of connection between the membrane and the cytoskeleton as what you see here, and this result in formation of microvasculation and membrane loss, and finally resulting in formation of spherocyte. Okay, so this spherocyte, when they go through circulation, when they reach the spleen, they are retained in the splenic cord because the red blood cell ideally has to transit from this splenic cord into the sinusoid, and you can see this is a very tight junction that it has to pass through. So the deformed spherocyte cannot pass through this tight junction, so they will be retained at the splenic cord and will be destructed, and this is where hemolysis occur.
With hereditary spherocytosis, like sickle cell disease, a genetic mistake causes the red cells to become misshapen as they travel through the blood system. In this case, they become sphere-shaped, like rigid basketballs. When these cells pass through the spleen, they are mistaken as damaged and get destroyed, causing anemia.
The second disease I'm covering in this mini lecture is keratin skin disease. Alright, so let's look at intermediate filaments. Intermediate filaments, they are primary component of cytoskeleton, and they are the most stable component of the cytoskeleton. Stable meaning they are strong here. Alright, therefore you find the intermediate filaments at or in the durable structures such as hair, scales, and fingernails. Here you can see a schematic diagram of hair fiber where you can see it is made of type 1 and type 2 of intermediate filament known as keratin. Alright, so the primary function of intermediate filament is to create a cell cohesion. Alright, and to prevent any fracture of the epithelial cell under tension. Don't worry about the epithelial cell structure as for now, because there will be a separate lecture covering those topics. Alright, here are the type of intermediate filaments, and type 1 and type 2 are known as keratin. So keratin consists of 54 unique genes in human, and they are expressed in cell differentiation and development. The keratin pair K5 and K14 and K1 with K10 are normally associated with epidermal keratinocyte. Okay, keratinocyte is major cell type in the skin epidermis. All right. Now let's look at the keratin disease. What I have here is a immunofluorescence staining of human epidermis showing the expression of keratin K5. Okay, so here in picture A, alright, so keratin K5 is in the basal layer, alright, and it is stained in the red color as what you see here, alright. And the keratin associated protein, which is known as profilaridin, all right, this is expressed in the outermost layer, all right, what you see in green color here. All right, now let's look at picture B. So in picture B, this is expression of K12 keratin, all right, in the epithelial cell line. Okay, so this is the cell line. So whatever red network you see, this is keratin K12 expression. And in picture C, you can see that this is a mutant, meaning the K12 keratin here is mutated one. All right, so this is due to a genetic defect or a mutation. All right, and you can see there's a complete collapse of the cytoskeleton. All right, you don't see a network of keratin K12 here, unlike picture B. It is all collapsed, and what you see here, it is aggregates of it. All right, so this is a type of keratin skin disease. Now what will happen when someone have this kind of mutation? Let's look the next slide. So here are some pictures showing you keratin disorder. A common feature in all this picture, what you can see is skin blistering. Alright, so it can be very painful and highly debilitating. Alright, explanation for each of these disorder is stated here. Alright, on the right side, and I don't expect you to remember what are the mutation in terms of which keratin type is affected. All right, so this is just provided as an example so you can relate what you have learned earlier with the clinical application or the disorders. All right. Right, so that's the end of this mini lecture. Thank you very much for listening. After this, there is a quiz for you to attempt. So make sure you attempt the quiz before you continue with the rest of the content for this help package.